This video is based on the process of protein synthesis. Protein synthesis has two major steps, that is transcription and translation. DNA and RNA are major components in this process. So we'll first compare DNA with RNA. We'll look at what is DNA and how it's different from RNA. We'll look at the codons and anticodons on RNA. Then we'll look at the process of transcription, and in part 2 of the video we will discuss translation. Now let's take a tour to the basics. Have you ever come across people saying that you look similar to your mom or dad, or that you have curly hair like your dad, or brown eyes like your mom? You might be wondering how the traits from your parents are transferred to you. Well, this is the role of DNA. DNA contains genes, which is the blueprint that tells the body which proteins or polypeptide is to be formed. DNA is a nucleic acid containing genetic instructions used in the formation of organisms, either plants or animals. The structure of the DNA molecule, which you can see in this diagram, is helical, or like a ladder which is twisted. The four nitrogenous bases form the rung or the steps of this helical molecule, while the phosphate group forms the backbone of this structure. DNA molecule is composed of two strands. Each strand is made up of building blocks called nucleotides. A nucleotide is composed of a phosphate group, a nitrogenous base, and a deoxyribose sugar. RNA is also a polynucleotide. The role of RNA, or ribonucleic acid, is to transcribe and translate the genetic information. RNA helps cells to translate instructions encoded in the DNA into actual proteins. Now let's compare RNA with DNA. DNA is double helical. While RNA is single-stranded. DNA contains deoxyribose sugar, while RNA contains ribose sugar. In DNA, the bases involved are adenine, guanine, thymine, and cytosine. And in RNA, the bases involved are adenine, guanine, uracil, and cytosine. So the thymine in DNA is replaced by uracil in RNA. Compared to RNA, DNA happens to be much more stable and longer than RNA. Now let's look into the process of transcription. Transcription is the process by which RNA is assembled from a DNA template. DNA contains a coding region, a promoter site, and a terminator site. A molecule of RNA polymerase binds to the promoter site. It moves along the DNA, separating the two double helix. Each now unpaired base will bind with a nucleotide which carries the appropriate base. In the synthesis of RNA, uracil is the nucleotide complementary to adenine. When the RNA polymerase reaches the terminator region, this process stops. DNA strands bind together once again as the new RNA molecule leaves to the cytoplasm. This RNA is a transcript of the message contained in the gene.